Thank you for the introduction, and thank you also for inviting me, both to the organizers and Bill and Melinda Gates Foundation, for bringing me the whole way from Denmark here to Washington, D.C. It's been a pleasure for me to join this meeting here, and my knowledge about TB has increased fold over the last couple of days, because I had to admit that my knowledge about infection diseases and TB is rather limited. As Jim said, I worked with oncology drug development, and also within the last 15, 20 years with drug diagnostic combination, this strategy which has turned out to be very powerful within oncology. I prepared a small agenda for you. I will talk a bit about the history about drug diagnostic combination. You heard a bit about it yesterday from Dr. Pasteur. I will talk about what is a companion diagnostic. I will define that based on what the regulator says. And I will also talk about some of the assay technologies that has been used and approved in relation to oncology drugs. I will talk a lot about the drug diagnostic co-development model, and specifically what can we achieve from this model, and then at the end try to summarize and come with a short conclusion. But as I said, my knowledge about tuberculosis is very limited. I am in fact a clinical pharmacist. I have received training in infection medicine and also tuberculosis, but as you can see, it's many years ago. So in order to prepare myself a bit for this presentation, a couple of weeks ago, I made a small brainstorm with myself, and you see some few of the outcomes here. It's not definitely a complete list, but I will highlight a couple of them all, and also specifically due to the way that I have listened to your discussion here over the last couple of days. And I think especially the question about drug resistance, that's a major problem within both areas. When we look at oncology patients, if you have an oncology patient in an advanced stage disease, in fact, no matter what type of treatment you are giving to this patient, it could be classical chemotherapy, it could be some of the new targeted compound, at some point of time, unfortunately, these patients will relapse and develop resistance. There are hope, as you also heard yesterday from Dr. Pasteur. There are, specifically in relation to the immune oncology treatment that we see now emerge, specifically the PD-L1 checkpoint inhibitors. Some few patients that respond to these drugs seem to have a prolonged response. So we are maybe at the beginning of curing cancer, but not any longer. So there are different challenges, but they are great within different areas. And if you look at differences, that's one of the things I would highlight here. That's availability of new drugs. This very, very different within these two areas. And we heard it again yesterday from Dr. Pasteur. There's a huge investment in oncology from the pharmaceutical company. It seems to fit their business model very well. It's unfortunately not the situation when we look at TB. And if we look at a new drug being approved, I saw an article from Nature Review Drug Discovery here a few weeks ago. They projected that here in the US next year, 1/3 of every new drug will belong to the oncology disease areas. Yeah. That's something that you could wish for within your therapeutic area. But I'm also happy to see what was presented yesterday there, that something is going on. But back to oncology. Why is this drug diagnostic strategy important? And why is it important to integrate molecular diagnostic when we talk about oncology? I think you get the answer from up here, disease heterogeneity. Cancer is a very heterogeneous disease, and we need to include molecular diagnostic if we should be able to come up with more and effective drug. We need to use the stratification strategy that we have used for a couple of decades. Drug work at a molecular level, and that's where we are going to find the solution to new and better drugs. And that's why molecular diagnostic plays such an important role in relation to developing new drugs within oncology. Also, talking a bit about the history, I think it's important that you also understand the background for where we are today within oncology. You heard also yesterday Dr. Pastor mention that the estrogen receptor, that was in the first biomarker that had importance and implication also in relation to treatment of patients. And in fact, the estrogen receptor, we have known that since the 50s. But in the 70s, there was a UK pharmaceutical company. The name was ICI Pharma. Today it's AstraZeneca. They developed the first selective estrogen receptor modulator, and that was tamoxifen. That was a drug that partly blocked the estrogen receptor. It has been known for decades, also before the 70s, that certain type of breast cancer tumors was dependent on stimulation by hormones. So going and partly block this was the idea from ICI Pharma that it would impact the growth of these tumors. And they did clinical development studies, as we do today. They were a bit more straightforward with phase 1, phase 2, phase 3 study. It seems to be different within oncology today. But if you go into the literature, you can find a phase 2 study, in fact published in 1976. And here the authors concluded from a phase 2 study where they have used trastuzumab for treatment of advanced breast cancer that there was a high degree of correlation between response to this new drug, tamoxifen, and that these patients was positive in this estrogen receptor assay. And the investigators here, they in fact suggested that such an assay should be used to select these patients for treatment with this new drug. So despite that this is more than 40 years old, this is in fact still the principles that we are working according to when we are discussing drug diagnostic strategy within oncology. We're trying to find a predictive biomarker that can say something about the likelihood of the individual patient to responding to the drug that we have under development. Despite that this, you can say, is likely the first example that we see within oncology, it was not the one that have the impact on what we have seen here within the last 15, 20 years. It was something that happened in the 80s with the discovery of human epidermal growth factor receptor 2. Some few years after, there was a scientist from California, from UCLA, his name is Dennis Lehman. He discovered that a certain group of women with breast cancer, in fact, has an amplification of the gene for this HER2 protein. And he also found out that this group of women had a much more poor prognosis than the rest of women with breast cancer. They have a much more aggressive disease. And in a publication that he published in Science in 1987, he in fact suggested that we should develop an inhibitor that could go and block this receptor protein, and then likely inhibit the downstream signaling, and then also make the tumor to shrink. This idea was taken up by a biopharmaceutical company here in US, also from California, Genentech, and the inhibitor became the monoclonal antibody trastuzumab. And when Genentech took this compound into the clinic, they have also developed a clinical trial assay. It was an immune histochemistry assay that looked for overexpression of the HER2 protein. We know that there's a very close link between amplification of the HER2 gene and overexpression of this HER2 protein. So the IHC assay, that would fulfill the goals in relation to be selective for the patient that they wanted to include in this study. And this hypothesis that they had, that it was specific this group of women that would respond to this drug, seemed to be true. The development that did up to the 90s demonstrated that it was specifically women that had overexpression of the HER2 protein that responded to this drug. And when they took the documentation to FDA, here we have proved that this drug is safe and effective. They took also with them the documentation for the assay. And in fact, on the same day, it was in the autumn of 1999, they got approval both for the drug and for the diagnostic assay. The assay, you can see, has the name of a HESEP test. And this, in fact, very important that it was a simultaneously approval, because this assay is a very important treatment decision tool for the individual physicians in order to find out whom to treat. The way that Genentech did this development in the 90s has in fact served as a kind of inspiration for a number of other drugs. So you can say the rest of the history is in fact a bit trivial, because they have all used this enrichment strategy specifically in relation to the clinical development. Of course, it's different compounds. You can see imatinib, crizotinib, and also one of the new pembrolizumab, the PD-1 inhibitors. But they have more or less used the same strategy. Of course, there are different analytical platforms used today. It's not any longer only IHC that is used, but I will come back to this. And if you have such a diagnostic assay linked specifically to a drug, as this IHC assay was linked to trastuzumab, it's a kind of companion to the drug. It's called a companion diagnostic. And FDA has defined this. Other regulatory bodies worldwide has defined this. We have just got new regulation in Europe 14 days ago, and it's a definition that looks very much like the one that FDA came out with 2-3 years ago. And they say that such a companion diagnostic is an in vitro diagnostic device that provides information that is essential for the safe and effective use of a corresponding product. And then they identify some sub-areas down there where they think such an assay is useful, and they say that it is for predicting efficacy, predicting serious adverse reactions of the drug, and also to monitoring therapy. In fact, I think I had to add here that in the beginning, and also when I started to work with these things, we didn't call it companion diagnostic. We called it pharmacodiagnostic assays or theranostics, and these names you might still hear them, but the regulators, they have decided we used companion diagnostics. I promised also to say a bit about the assay technology that are used in these companion diagnostics, which in fact has been approved by FDA and also other regulatory bodies. It is so when you have this parallel approval by FDA, it's normally it's CDER that do the drug approval and CDRH that approve your assay. And all these assays here, they have been improved by something called a PMA. That's the highest documentation level that is required by CDER. And here you can see the technology. It's immunohistochemistry, just as we talked about here in relation to trastuzumab, and you can also see some of the assay and some of the drugs mentioned there. If you look for gene amplification, gene deletions, or gene translocations, you can use in situ hybridization, or you can also use real-time PCR, but real-time PCR in relation to companion diagnostic is mainly used for detecting mutations, and the same is with DNA sequencing. In fact, when it comes to next generation sequencing, the first companion diagnostic based on this technology was approved just before Christmas. So it was the first time, at least using this parallel drug diagnostic strategy, that this was happening. I know there are other examples, but this was the one that went to a PMA approval. I promised also to talk about the drug diagnostic co-development model, and we made a flowchart some years ago in an article we published in Frontiers in Oncology. And this flowchart you see here is a kind of textbook version, because also, as we heard from Pasteur yesterday, and as you will see later on in my presentation, very few, in fact, phase 3 studies are done nowadays within oncology. But I will not highlight every single aspect in this sequence here. Up here you see the drug development sequence, and down you see here the sequences in relation to development of the assay. But just look at some few important aspects. And the important aspect, this is the biomarker hypothesis, that you're in the beginning of the drug development project to come up with a strong biomarker hypothesis. And when I talk about a strong biomarker hypothesis, I talk about the link between the biomarker status and the outcome of the drug. It could be efficacy, it could also be safety. Such a biomarker hypothesis is normally generated based on the basic research that you perform here during the discovery phase, or it can come even earlier based on academic research. And if you have such a strong biomarker hypothesis, the next step is to develop a prototype assay. A prototype assay that you then need to test out up here in the early clinical development in order to look for a predictive potential of your biomarker. And if you find such one, the next step to do is to establish the cutoff for your assay. And the cutoff for your assay has nothing to do with the analytical sensitivity or specificity. It's related to the clinical. It's the cut point or the value from your assay that split your patient population into likely responders, likely non-responders. And it can be very challenging to find such a cut point or cutoff for your assay due to the fact that you have relatively few clinical outcome data up here. It's from phase 1A and B and maybe from phase 2A. If you are able to select such a cutoff, the next thing is analytical validation, and this, as also I can hear from some other publications, this is definitely, or presentation here, is definitely not a trivial procedure, and it's in fact a very, very important procedure in relation to a companion diagnostic. It doesn't matter that you have a strong biomarker hypothesis down here and you have proved that it has predictive potential if you cannot trust your assay. The assay has to be robust, it has to be reliable, because you based your treatment decision based on the outcome of this assay. So you need to be able to trust it. And it's not only that you need to analytical validate your assay, you need also to clinical validate your assay, and that's the next step here. And you clinical validate your assay based on the outcome data that you find here from phase 2 or sometime also phase 3. Here you demonstrate that in fact the group of patients that your assay select is the one that also do respond to your treatment. And if then your drugs prove to be safe and effective, and you have sufficient analytical and clinical validate your assay, you would likely be obtaining a regulatory approval simultaneously, as we talked about before, and it is very important. Assay need to be available at the same time as the drug is available. It is an important treatment decision tool, and the physician that prescribing these new drugs need it. When we look at the clinical part of the drug diagnostic development strategy, there's one study design that has been used over and over again, and that is the enrichment study design. And you can find a number of different variations of this design, also basket trials and so on. I present only the simple version here for you today. But this study design is in fact built on the traditional randomized clinical study. You put in an extra step. You see the testing here with the companion diagnostic assay. You can call this is the enrichment step, because you enrich your study population with biomarker positive patients. And then you start your randomization to the new drug and standard treatment. And of course, such a study design is only useful if there is a very strong link between biomarker status and the outcome of the treatment. Otherwise, you should forget it. It will not be ethical justifiable to do such a design. There's other designs that then can be used. And this was the design that Genentech used to prove the efficacy and safety of the drug, and also at the same time clinical validate their companion diagnostic assay. And it has been used for a number of other drug diagnostic projects up till 5-6 years ago. 5-6 years ago, we certainly saw a simpler design. In fact, as simple as it possible could be. Also a design that was able to gain approval of a drug. And this was the open, non-randomized enrichment study design, as you see it down here. So again, of course, we have a protocol that defines the inclusion criteria, and you test your patient, and then you treatment and see what happened. And within oncology, we have something called the RECIST criteria that we used in relation to define efficacy, the endpoint in the clinical study. And it is response rate, as we also heard yesterday. It can be progression-free survival when we are talking about advanced stage cancer. And the first drug, in fact, to be successful using this very, very simple clinical development strategy, this was crizotinib. It's a multi-kinase inhibitor. It inhibits both ALK, it inhibits MET, and also ROS1. And Pfizer, that developed this drug, decided to develop it initially for non-small cell lung cancer for patient has an LM4 ALK rearrangement. It's approximately 5 to 7% of the patient that has non-small cell lung cancer that have this specific gene aberration. And you test these patients with an in situ hybridization assay in order to find them. That was at least what Pfizer did. And in fact, 255 patients was sufficient to demonstrate efficacy of this drug in 2 open-label, simple studies, enrichment studies. And when I saw this first time, I said, this cannot be true. I have been gone to the old school, as also Pastor talked about yesterday. If you want to have a drug approved by the regulators, it is 2 phase 3 studies, randomized phase 3 studies, independent studies, and then certainly Pfizer could get away here with 2 simple open-label studies with relatively few patients. But the explanation, you heard it also yesterday from Pasteur, these drugs are much more effective than current standard therapy. And if you look at this was at least one of the explanations he gave, and if we look at crizotinib and compare it to chemotherapy, this has a response rate that are 3 to 4 times higher. The response rate here in these studies was around 60% or a bit above 60%. And if you look at standard chemotherapy, it's around 10-50% if you're lucky, if you have an advanced stage non-small cell lung cancer patient. Not only was this drug more effective, it was also more safe. You know, all chemotherapy has a number of serious and sometimes even life-threatening side effects. And finally, we are within oncology, as we also heard yesterday. The barrier are lower. We are talking about life-threatening diseases. The life expectancy for these patients here can be counted in months. And then we are within an indication also where the population of these specific patients with this specific gene aberration is small. But this was not the only example. As you can see, I have listed some other examples that has been approved by FDA and also all other regulators globally. And some of them used only one study to have the drug approved, and this was ceritinib and also crizotinib. And you can see here it's developed also for the ROS positive patient population. So you can say, if you look at the first 4 examples there, oh yes, this is only non-small cell lung cancer. It's only tyrosine kinase inhibitor. No, just before Christmas, I think it was on the 19th of December, rubicarib was approved for advanced stage ovarian cancer. It's a PARP inhibitor, so it has a completely different mechanism of action than the tyrosine kinase inhibitors. And it was intended for patients that has BRCA1, BRCA2 mutations using next generation sequencing. It was the first time we saw that in such a drug-diagnostic combination strategy that this was approved as an analytical platform. And again, we are above 50% in response rate for these patients. So no doubt that you are able, if you can pre-select out your patient based on molecular characteristic and have sufficient knowledge about the drug mechanism of action, you have the possibility of increasing the efficacy rate of your drug using this drug diagnostic strategy. And I made also a small survey which was published in Trend in Molecular Medicine some 1.5 years ago. I just went to the FDA's drug database, looked at oncology drug that has been approved for advanced stage cancer over the last 15 years, so it was after the approval of trastuzumab. And then I looked at the response rates and divided them into two groups. Doctor has used a drug diagnostic strategy for the development, and another group where this was not the situation. And as you can see here, the upper part of this table there, this is the drugs that has been gone through the drug diagnostic strategy. And if we look at the response rates, it's from 34 up to 80%. And if you look down here where they have used a more traditional all-comers strategy, the response rate is somewhat lower. Of course, this is not a scientific comparison, just a simple survey, but despite of this, it points in the right direction. It points that this strategy, by stratifying the patients according to their molecular diagnostic when we are within quality, do matter. Where it also seems to matter, that is in relation to the success of such a drug development program. There's a group of researchers from Canada, from the University of Toronto, that had looked into these things. They have published a couple of papers, and I know that they have more papers on the way, and I will look forward to see them, because it's a way also to evaluate what this strategy can do. And they looked at melanoma, and they looked at non-small cell lung cancer. They looked at the cumulative success rates from passing from phase 1 until phase 3, if there was a phase 3 study included, and have the drug regulatory approved. And as you can see, if you use the all-comers strategy here, the traditional one, the success rate, when you start with a drug in melanoma and have it start in phase 1 and have it approved, is approximately 6%, and a little bit higher for non-small cell lung cancer, 11%. But if you use the drug diagnostic strategy, you can see the success rate increase morefold. Of course, you should not put too much emphasis into these data to the fact that they are still relatively few. Over here, there's a number of data points behind these figures here, but over here, there's only a few. But again, I'm an optimistic type, so I think this point in the direction that we are using a strategy that seems to work. But again, it is a few data points that these figures are built on. What about the cost? Cost is an important aspect when we are talking about drug development, and it's something that has been discussed a number of times. A couple of years ago, there was an institution here in US. They estimated the average cost of drug development to be $2.6 billion, an enormous amount of money. It was discussed also in New England Journal of Medicine what to include there. But if we take this $2.6 billion, as you can say, the kind of fixed item there, and then compare it to the drug diagnostic strategy. And again, our colleagues there at Toronto University, they estimated the drug development cost for crizotinib, and they came up with a figure of $500 to $600 million, significantly lower. So also, with this respect, it seems like the drug diagnostic strategy have something very positive to offer. Come to the conclusion, and today 20 drugs has been developed using this drug diagnostic strategy and being approved together with a companion diagnostic. And if you plan to develop a companion diagnostic, there are a few key requirements that is important to remember. A strong biomarker hypothesis, that's of course a must, and then analytical and clinical validation. If you forget it, if you work within drug diagnostic strategy, I'm absolutely sure that the regulators, they will remind you that this is very, very important, and I completely agree. This drug diagnostic strategy seems to offer some advantages, both in relation to efficacy, seems also to offer some advantages in relation to success rate of a drug development project. There might also be cost reductions, and in fact also it might also impact the time that you spend on such a drug development project. In fact, for crizotinib, I know that was a bit special, but it took only 4 years from the discovery of the ELM4 gene translocation and until they have a drug that was approved and could be of benefit to the patient and also to Pfizer that developed and could start to have some return on their investment. So the last question we have to answer here is, of course, can this drug diagnostic strategy we use in oncology, can this be translated into other disease areas? I think yes, but I will not say anything about TB. But the reason why I said yes is that we have seen this strong link within other disease areas. But again, it is mainly within oncology that we see this very, very strong link between the biomarker status and the outcome of the drug, at least so far. If you think what I've told you here is interesting, I've been involved in a number of projects. I was also involved in being an editor for a specific issue on frontiers in oncology that came in 2014. This can be downloaded free of charge. It has to do with drug diagnostic co-development within oncology. A number of researchers has contributed to this. And very recently also, I do a bit promotion here, we have a couple of publications coming out. They are open access publications, came in December. This one from Annals of Translational Medicine, and this one from a Cell Press journal here. And they can be downloaded free of charge. And then, just to come back to what Jim said here in the beginning here, that we should value biomarkers. I will give the last word here to the current president of ASCO, Daniel Hayes. He's been one of the pioneers within this area. He gave an interview in Expert Review in Molecular Medicine a couple of years ago, and here he said that a bad tumor biomarker test is as bad as a bad drug. I would say that a good tumor biomarker is as good as a good drug. So it's to your discussion about what is best, you know. I think they are equal important. And today, when we are talking about oncology, these assays, they are the gatekeepers. They are the one that decide if a patient should be treated with a drug. I know that any physician has his free prescription right, but it is also very much linked to reimbursement. So I don't think that any doctor will prescribe a drug without having done these tests. Thank you very much for your attention, and thank you very much for inviting me.